the messiness associated with it. Right? There's a lot of mess and a lot of scientific mess as well. I mean, you would have figured all of this out. Separately, at some point, the actual structure of penicillin was discovered. And then what it attacks was discovered. And that brings me to how antibiotics work, right? So penicillin is something that attacks something called peptidoglycans. These make up the cell wall of the bacterium.
Okay.
And these, so it goes and attacks this complex of a protein and a starch molecule, something like that, and goes and cuts that, and therefore the cell becomes weak. Water rushes into the cell. So all our cells have a delicate pressure, osmotic pressure, right, with salt concentrations, which is why you can never inject just pure water into your bloodstream. And as a result, so these cells sort of explode. So various antibiotics either make bacteria stupefied and sort of inactive, or they kill them. And they do this by attacking various parts of it. You can destroy the cell wall. You can destroy the cell membrane. Think of, trying to injure another person, right? These are almost like physical attacks.
Right.
And then you have poisons of various sorts. You'll have essential enzymes, essential proteins that are crucial to the respiration of that bacterium, to the metabolism, how it does digestion. So you block any of those systems which are critical to the life of that bacterium, you can destroy that bacterium.
So what are these antibiotics by itself?
So they're small molecules.
They're
so
small
they're
molecules?
small organic molecules.
Okay.
So there'll be a benzene ring, there'll be some other ring, some CH3 groups here and there, some nitrogen here or there. The sulfur drugs have a sulfur atom working with some oxygen atoms. But these are largely small chemical molecules.
Sure. Now, we get that from your story that penicillin was found or discovered because of Fleming's, let's say, hygiene issues of leaving his plate out without cleaning it.
By the way, the plate was an agar plate of bacterium, not an eating plate.
Okay. Nonetheless, how do other antibiotics get discovered?
So after this, a lot of work was happening. So what you see is between 1949 or so, or 1947 and 1977, every year there were multiple antibiotics that were discovered. Many of these were variants of existing antibiotics. So you make it slightly more effective, you modify something there, something might have a slightly lesser side effect, something might just be more effective. Some, like penicillin, which was naturally made, in a minority of people triggered allergic reactions. So you have something purer, less allergic profile, etc. Right? But the last in these antibiotics, some are considered broad spectrum, so they'll tackle a wide range of bacteria, and some are narrow. They'll only hit certain types of bacteria. And so even in our diagnostic process, when people are not sure what kind of infection you have, they'll hit you with a broad spectrum.
Oh, right. Okay.
Right?
Yeah.
And you can separately debate misuse, overuse, all of that. And only once they know, okay, you have paratyphoid. Okay, I have a specific drug that will work best against paratyphoid. Right? So that's when you can use a specific drug. And in this, I don't want to get into the details. In bacteria, you classify them as gram positive and gram negative, basically based on how an old stain called the gram staining method. If the stain sticks to the bacteria, it's positive. It doesn't stick to the bacteria, negative, whatever. It's some way of classifying. For gram negative bacteria, which include a lot of important diseases, we haven't had a new antibiotic since 1962.
Right, okay. That's a long time back. Yeah.
And like you earlier mentioned, with antibiotics, it is a cop and robber game, right? So there is resistance that is built. So you bring a knife, then somebody brings a gun to that knife fight, and then you bring a machine gun to that gunfight. Yeah. That is a process that happens. And in bacteria, it happens in two ways, right? So imagine there are 1 million bacteria. You hit that spot with antibiotics. It kills 99.9% of it. But there'll be a tiny part, 0.01% of those bacteria, which naturally just have something right or wrong with them, which makes them resistant to this antibiotic. And therefore, only those survive. And then they multiply in no time to form the full population.
So you use Harpic every day.
Yes. And even if you use Harpic, you actually are breeding these bacteria that are extremely resistant. The other way is actually they can bacteria have sex in certain ways. So they can actually transfer genes from one bacterium to